Welcome to Lecture 18, where we will go over the endocrine system. There are many factors that determine the effect of a hormone within a body. This will in turn also have an effect on an animal's behavior. These factors include the concentration of the hormone in the body, the presence of the hormone in a cell, and the response of the cell when the receptor binds to a hormone. A hormone is defined as a secreted molecule that travels in the blood to cause a response in specific cells or tissues. It is important to note that target cells must have the proper receptor in order to be able to respond. The communication and regulation of body processes is controlled by two main systems, the endocrine system and the nervous system. The endocrine system involves chemical signaling using hormones, while the nervous system involves transmission of signals through a network of neurons. Many times these systems work in highly coordinated mechanisms with each other. There are many different ways that animal cells can communicate with each other. We will discuss 5 of the main signaling pathways. The first pathway we will discuss is endocrine signaling. Endocrine signaling involves the secretion of hormones into the bloodstream or into the hemolymph. This will eventually reach the target cells or tissues. Endocrine signaling pathways are important in maintaining homeostasis, in regulating processes such as growth and development, and also in regulating reproductive changes that occur at maturity. Cells can also communicate with each other through paracrine and autocrine signaling. In both of these types of signaling pathways, local regulator or signaling molecules are released and they can act on neighboring cells in the general vicinity, as we see in paracrine signaling, or they can actually act on the cell that is producing the regulator molecules themselves, as we can see in autocrine signaling. This type of signaling is important in regulating various physiological processes, such as blood pressure, nervous system function, as well as reproduction. The final way that communication can occur between cells is through synaptic signaling or neuroendocrine signaling. In synaptic signaling, neurons will secrete neurotransmitters into a synapse. These neurotransmitters will then bind to receptors on the target cells, either muscles or other neurons, to cause a response. Synaptic signaling is important in memory formation, cognition, movement, as well as detecting sensations. Neuroendocrine signaling is a joining between synaptic signaling and endocrine signaling. In neuroendocrine signaling, neurosecretory cells will secrete neurohormones. These will enter into the bloodstream to have a response in target cells or tissues. This is involved in regulating the endocrine signaling pathways. Please review the images below to get an idea of what is involved in each of these signaling pathways. In general, there are 3 main classes of hormones: polypeptides, steroids, and amines. These hormones can also be classified based upon their solubility as well as their base structure. Polypeptides are generally considered to be water-soluble or hydrophilic. Steroids, on the other hand, are considered to be hydrophobic and are lipid-soluble. Amines can be either or depending upon their base structure. For instance, epinephrine is considered to be a hydrophilic hormone, whereas thyroxine is considered to be hydrophobic. Hormones will take 2 basic pathways depending on their solubility. When we consider any of the hydrophilic hormones, these hormones can bind to receptors on the cell membrane. This is because they are hydrophilic and cannot pass through the cell membrane on their own. When the hormone binds to the specific receptor, it will initiate a signal transduction pathway in the target cell to cause some sort of cellular response. This cellular response can include activating an enzyme, uptaking or secreting a specific molecule. It can also involve cellular changes, altering the levels of gene expression, and many other types of cellular responses, such as an increase in cell division, for instance. Take a look at the diagram to the right to understand the general pathway that a hydrophilic hormone would take. Note the presence of the receptor protein on the membrane of the target cell. An example of a hydrophilic hormone is epinephrine. When stressed, the adrenal glands that reside on top of the kidneys release epinephrine or adrenaline. This has a response on many tissues, including the liver. The epinephrine pathway involves what is termed G-protein coupled receptors. These are specific receptors that bind to the hormone that are held in close association and work with G-proteins. This will then activate other membrane enzymes such as adenylyl cyclase that we see here. These pathways also typically involve secondary messengers such as cyclic AMP or calcium to help amplify and coordinate the cellular response. In the epinephrine pathway, epinephrine will bind to the G-protein coupled receptor in the membrane of the target cell. This will activate the G-protein, which will slide along the membrane to activate adenylyl cyclase. Adenylyl cyclase is a membrane-bound enzyme that's responsible for converting ATP into cyclic AMP, which is a secondary messenger. When cyclic AMP is produced, this will then activate protein kinase A. In the epinephrine pathway, this will inhibit the synthesis of glycogen or storage of glucose molecules. It will also promote the breakdown of glycogen to release glucose into the bloodstream, thus increasing the amount of potential energy available to cells and tissues. We're now going to take a look at the general pathway involved in hydrophobic hormones. Remember that hydrophobic hormones can easily pass through the plasma membrane. In some instances, the hormone can also pass through the nuclear membrane. As a result, the receptors for these hormones are going to be interior to the cell, either in the cytosol or possibly within the nucleus. The hormone will, in general, pass through the cell membrane and bind to a receptor in the cytosol, or as we see in the diagram to the right, it may bind to its receptor in the nucleus. Once the hormone binds to its receptor, regardless of where it is, it forms a hormone receptor complex, or HRE. This complex will then move into the nucleus, if it isn't already there, to alter gene transcription to cause a cellular response. In general, these pathways take much longer than what we've seen for hydrophilic hormones. Steroids such as estrogens and testosterones can easily enter into the cells and typically cause a cellular response through the activation or alteration of gene transcription rates. We're going to take a look at the specific pathway involved in estradiol. Estradiol is considered to be a type of estrogen. Estradiol, because it is hydrophobic, will easily pass through the plasma membrane, where it will bind to its receptor in the cytosol. This will form the hormone receptor complex that will then enter into the nucleus. Once it enters into the nucleus, it will alter gene transcription rates. Specifically, it will upregulate the production of mRNA for a protein called vitellogenin. Vitellogenin is an important protein that is used for the production of yolk. When estradiol enters into liver cells, it will cause an increase in the production and secretion of vitellogenin proteins. These proteins will then circulate into the blood to reach the reproductive organs, which will then be harvested for deposition in the yolk. Although we see that the interaction between a hormone and its receptor is very specific, a hormone can elicit various responses simply due to slight changes in the receptor itself or in any component involved in the signaling pathway. For instance, epinephrine plays a very important role in the fight-or-flight response. However, the secretion of this one hormone will trigger a range of coordinated responses in the body. These include a general rise in body temperature or excessive sweating, an increased heart and respiration rate, as well as an increased metabolic rate, tunnel vision and selective hearing, tightening of muscles, dilated pupils, the constriction of blood vessels, as well as a slower digestive function. We also see the maintenance of goosebumps or the constriction of erector pili muscles. Specialized cells within the endocrine system are called endocrine cells. These cells can be present simply as a group of aggregated cells that exist within an organ, or they may actually form their own endocrine gland. Take a look at the diagram below to see the overall endocrine glands that are present within the human body. Note that they can be present in multiple organs, such as the adrenal glands, as well as within the brain with the pineal gland and hypothalamus, and they also are present within the gonads. Also recall the difference between endocrine and exocrine glands. Endocrine glands secrete hormones into the surrounding fluid or into the bloodstream, while exocrine glands, like sweat and salivary glands, contain ducts. These ducts will secrete the substances produced by the exocrine glands directly onto the body surface or onto the surface of a cavity. There are two main pathways that are involved in the regulation and coordination of the endocrine system. These involve an endocrine pathway or the neuroendocrine pathway. In the endocrine pathway, endocrine cells will respond to stimuli, both internal and external, by secreting a hormone that then enters into the bloodstream to reach the target tissue. This hormone will then bind to specific receptors, as described in the previous slides, to initiate a cellular or physiological response. An example of this is the secretin pathway, or epinephrine pathway. In the simple neuroendocrine pathway, sensory neurons will detect a stimulus. They will then respond by sending nerve impulses to a neurosecretory cell. These neurosecretory cells will then produce and secrete neurohormones. These neurohormones are then released in the system to bind to specific receptors on their target tissue or target cell. This will then cause a cellular or physiological response. An example of a hormone that is controlled by a neuroendocrine pathway is oxytocin. We will now take a look at the differences between these two types of pathways, specifically looking at the pathway of secretin and oxytocin. Secretin is an example of a simple endocrine pathway. The stimulus in this case is a low pH in the duodenum. When the pH in the duodenum drops, S cells of the duodenum release secretin. These will then circulate through the bloodstream to then reach their target cells of the pancreas. When the hormone binds to the receptor on the target cells, pancreatic cells will produce bicarbonate. This will then travel through ducts to then be released in the duodenum. This will cause an increase in the pH. This is also termed a negative feedback loop. As bicarbonate is released and the pH increases, this would then cause a reduction in the production of secretin. We'll now take a look at the neuroendocrine pathway as evident in the oxytocin signaling pathway. The stimulus in this case is the suckling of an infant. This will then cause activation of the hypothalamus gland, specifically neurosecretory cells in the hypothalamus. These neurosecretory cells will be activated to secrete the hormone oxytocin. Oxytocin will then be released from the posterior pituitary to then travel in the bloodstream and circulate throughout the body. These will then reach their target cells, in this case the smooth muscle mammary glands. When oxytocin binds to the smooth muscle in the mammary glands, this will then cause the contraction of the smooth muscle to cause milk release. This is also considered to be a positive feedback, where the release of milk will cause further stimulus of the signaling pathway, eventually until the suckling infant stops and is satiated. Feedback loops, negative and positive, are very important in linking the overall response of the pathway back to the initial stimulus. In a negative feedback response, as we've seen in the previous slide, the response serves to decrease or inhibit the initial stimulus. This is very common in regulating systems because it is important in restoring the initial state. The example is the slide we've seen before with the release of secretin. The release of secretin will cause the release of bicarbonate that will help to increase intestinal pH and stop the release of secretin. In a positive feedback loop, we have the opposite. In this instance, the response will serve to increase or enhance the initial stimulus. These are less common, but they are also important in providing control for the body. An example, as we've seen in the previous slide, was the oxytocin signaling pathway. In this pathway, the release of milk causes an increase in suckling, which is the stimulus. This will cause a subsequent increase in oxytocin release until the suckling ceases. In many animals, the endocrine and nervous system work in such high coordination that it can be termed the neuroendocrine system. For instance, in invertebrates, the molting process or ecdysis begins when neurosecretory cells in the brains produce PTTH. PTTH stands for prothoracicotropic hormone. This hormone will travel within the hemolymph where it reaches the prothoracic gland. This will then cause a response in the prothoracic gland cells to cause the release of ecdysteroid. Ecdysteroid will control the process of metamorphosis. It will determine when larval tissues undergo apoptosis and the adult cells will take over. These cells will develop into adult structures such as legs, eyes, antennae, etc. In addition, juvenile hormone will modulate the activity of ecdysteroid. Juvenile hormone is generally present in high levels during the larval stages. However, a subsequent drop in juvenile hormone will induce the process of metamorphosis. Remember, metamorphosis is a stage in holometabolous insects where you get the complete conversion of larval tissues into the adult form, and this involves the formation of a pupal stage in order for metamorphosis to occur. This diagram depicts the relationship of PTTH, juvenile hormone, and ecdysteroid in the development of a caterpillar. The balance between these hormones will determine the difference between when a larvae progresses through successive molts or when it needs to form a pupa. This will then lead to metamorphosis that will then lead to the emergence of the adult form. Ecdysteroid is released episodically, which means that it is released in pulses within the body. These are highly controlled and regulated through the neurosecretory cells. When there is a high level of juvenile hormone, with these ecdysteroid pulses, you will get the successive molting between larval stages. Pupation is generally induced when there is a distinct drop in juvenile hormone with a consistent pulse of ecdysteroid. In addition, emergence from the pupa to the adult stage is typically controlled by ecdysteroid in the absence of juvenile hormone. In general, high levels of juvenile hormone suppress the metamorphosis of the larvae into the adult stage. In vertebrates, the hypothalamus initiates neuroendocrine signaling. This is typically in response to stimuli from the body or external environment. The hypothalamus will then send signals to the pituitary gland. The anterior pituitary is an endocrine gland that produces and secretes hormones in response to hormones released from the hypothalamus. Rather, the posterior pituitary contains axons from the hypothalamus. This will then lead to the secretion of neurohormones.